In this tutorial, we are going to look at cell division, and specifically, we're going to focus on a type of cell division called mitosis. Now, mitosis happens in somatic cells. A somatic cell is a body cell, and the purpose of mitosis is for growth and maintenance. When we talk about cell division, we really talk about it in the realm of what's called the cell cycle. The cell cycle is the time period from which a cell first arises until it divides. And there are three main stages of the cell cycle. The first stage is called interphase. The second stage is what we refer to as mitosis. And the third stage is cytokinesis, in which the cell actually splits into two daughter cells. Now, here in this photo, you're actually seeing a summary of the cell cycle. This very first cell is where we begin. And this is in a stage called interphase. Interphase is a time in which the cell is undergoing its normal everyday functions and it's preparing to divide. Once that preparation has taken place, the cell enters mitosis. Now, by definition, mitosis is the division of the nucleus, and the cell will go through four substages during mitosis. The first is called prophase, and these two cells are prophase. Next is metaphase, which the chromosomes line up down the center. Then we enter what's called anaphase. These two cells are in anaphase. And the last stage is telophase. Telophase is when the cell or the new nucleus begins to form. The nuclear envelope forms around those chromosomes and the cell is just about to divide. So collectively, prophase, metaphase, anaphase, and telophase is what we refer to as mitosis. So all of these here would be our second stage of the cell cycle, called mitosis. And the very end of the cell cycle, and it occurs right at the tip end of mitosis, is when the cell actually splits. The cytoplasm pulls apart and you end up with two daughter cells. We call these two cells daughters. They're actually identical to the parent cell that we started with in interphase. This is cytokinesis. Cytokinesis is the actual division of the nucleus. So we're going to progress through each of these stages of the cell cycle. Here we're seeing a cell in the very first stage of the cell cycle known as interphase. Now, interphase is the longest stage of the cell cycle, and when you view tissues under the microscope, 99.999% of those cells are going to be in interphase. It's actually very difficult to catch a cell undergoing mitosis. Most of the time when we look at mitotic phases under the microscope, we're viewing embryonic cells. These are going to be cells that are dividing rapidly, so it's easier to catch a cell in a stage of mitosis. Again, most of the cells will be in interphase. So what we're looking at here is a picture of a microscope slide, and we're seeing a cell in interphase. So I want to bring your attention first off to the nucleus. So this whole thing right here is the cell that we're focusing on, and you can see right in the middle is going to be that nice big nucleus. A key characteristic of interphase is that you will see a cell with a nucleus and the nuclear envelope will still be intact. DNA is also in the form of chromatin. Now, there are three substages of interphase. The first is called G1, which stands for gap 1, but I want you to think of that G as growth, because this is a period in which the cell is growing in size, it's getting larger. We'll also start to see organelles begin to replicate. If you look over in the photo to the right, you'll notice that the centrosome has replicated in this drawing. here's a centrosome and it's duplicated, so there's another one right here. Now remember, the centrosome is actually made of two parts: the centrioles, which produce the microtubules, and the pericentriolar material, which is pretty much the organizing center for producing those microtubules. During cell division, the centrosome plays an essential role. That centrosome produces what we call spindle fibers. Spindle fibers are specialized microtubules that are going to play a role in moving those chromosomes. As the cell passes through those substages of interphase, G1, S, G2, it will soon enter mitosis. Now, by definition, mitosis is the division of the nucleus. Mitosis has 4 substages that result in the production of chromosomes, the movement of those chromosomes, and eventually the separation of the nucleus. So you'll end up with a cell with 2 nuclei. Here we've progressed to the very first stage of mitosis, which is known as prophase. So we are looking at prophase. Now, in prophase, chromosomes will begin to form. The nuclear envelope starts to break down and separate out. And these spindle fibers that we mentioned earlier that the centrioles or the centrosome is going to produce, the spindle fibers form. Here in the photo to the left, you're seeing a drawing of these events taking place. Notice we have the breakdown of the nuclear envelope. Inside, we've got the formation of chromosomes. And then outside the nucleus, you can see the spindle fibers starting to form. Now you'll notice this diagram calls them microtubules, and they are microtubules. That's actually what they're made of. Usually in terms of cell division, we refer to them as spindle fibers. So I'm going to name them spindle fibers here. Now, under the microscope, we really can't see all of these little details taking place. But what we can see is the presence of chromosomes and no nuclear envelope. Over here to the right, you're seeing chromosomes on the inside of that cell. This is my cell right here. You can see that little jagged edge. That's the plasma membrane that we're looking at. Here in the middle would be the cytoplasm. And then right here in the very center are these little rod-like structures. These are the chromosomes. Now, in textbooks, you usually see chromosomes shown as little X's. And if you look at the drawing to the left, they look like little strings of X's. The reason they look like X's is because previously, during the S stage of interphase, the DNA replicated. So when the DNA replicates, you end up with 2 identical copies. And these 2 identical copies are connected to each other. So they often look like little X's. And as mitosis progresses, as we continue to move through the different phases of mitosis, these little duplicate copies will separate out so that each new cell has its own set. For identification purposes, you're looking in prophase for a cell that has chromosomes, little rod-like structures, and no visible nuclear envelope. Remember, that nuclear envelope has broken down. Next up is metaphase. So what we're seeing here after prophase is metaphase. Now, metaphase is pretty easy to identify. Once your chromosomes have formed and the nuclear envelope has separated out and dissipated in prophase, there's some room for these chromosomes to start moving around. The spindle fibers are going to start to attach to your chromosomes and organize them. They're going to line them up in a nice, neat little line down the center of the cell. A lot of times it's referred to as the equator of the cell. So when we're identifying metaphase, all we really need to do is just pick out a cell that has a line of chromosomes directly on that equator down the center of the line. Here in the drawing, you can see the centrioles have migrated to opposite sides, there and there. You can see the spindle fibers, now identified as spindle fibers, attached to your chromosomes, and then you see the line of chromosomes right down the center or the equator of that cell. Over on the right, you can see a little bit about those spindle fibers, these little lines going through or going to those chromosomes. And this little starburst area right in here, this is where your centrioles would be. A lot of times these are referred to as asters, little starburst-like structures. And then, of course, right down the equator of the cell, you've got those chromosomes. So in metaphase, your chromosomes line up down the middle. Once your chromosomes are lined up in the middle of the cell, nice and organized, the cell is ready to enter the next stage of mitosis called anaphase. So next up, and what we're seeing here, is anaphase. Anaphase is when the chromosomes begin to separate. Now these centrioles, which are holding the spindle fibers in place, begin to pull on those spindle fibers. And since the spindle fibers are attached to each chromosome, remember those, they look like X's because they're duplicated, they're replicated parts. Since the centrioles are attached to each of those, when they begin to contract and the spindle fibers pull apart and shorten, those little X's begin to pull apart and separate. So anaphase is the actual separation of your duplicated chromosomes. We refer to these duplicated parts as sister chromatids. So when you're reading about this, you might see a description that says the separation of sister chromatids. And what that means is that your replicated chromosomes begin to pull apart. Over here on the right-hand side, you can see that the shape of our cell has actually changed a little bit. The shape of the cell looks a little bit more like an oval. See how wide it has gotten? And then you can see two darkened areas on either side here, and then here's the other darkened area, and then in the middle is this clear space. That clear space is the separation of those sister chromatids or those duplicated parts. So you're seeing a side come over to the right, a side of the chromosomes come over to one way, and then the side of the other chromosomes going in the opposite direction. Now, another key characteristic that happens in anaphase is that we begin to form what's called a cleavage furrow. Now, a lot of times in early anaphase, and that's actually what we're looking at here, the cleavage furrow is not very visible. A cleavage furrow is an indentation, a little dip at the top and the bottom of the cell. It would happen right around in this area, right in here. I'm going to circle that, and right around in here at the bottom. Now again, in anaphase and using a common light microscope, this cleavage furrow is not very visible to us, but it is gradually beginning to form here in anaphase. So if you ever have a little test question that asks, when does the cleavage furrow begin to form? Your answer is going to be anaphase. Even though it's not really detectable, it is beginning to form in anaphase. And by late anaphase, and especially telophase, which is our next stage of the cell cycle, that cleavage furrow will be very visible. We have now arrived at the last stage of mitosis called telophase. So what we're seeing here is telophase. Now, telophase begins to happen at the very end of anaphase with the deepening of the cleavage furrow and the formation of a new nuclear envelope. In the photo to the left, you can gradually see this nuclear envelope beginning to reform, and also you can tell how deep that cleavage furrow is starting to become. That's because we're gradually progressing and getting closer to what we call cytokinesis. Cytokinesis is the actual last stage of the cell cycle in which your cytoplasm actually splits. In the photo to the right, you're looking at a cell in telophase. Notice how big that cell is. I'm going to circle the whole thing, and you can see that cleavage furrow is quite deep, and a new plasma membrane is even beginning to form between your two cells. There's a clear cleavage furrow at the top and even at the bottom, with that new plasma membrane beginning to form to separate your cells. Now, under our light microscopes, we can't really see that nuclear envelope beginning to reform, but it is. and your DNA is still going to be in the form of chromosomes in telophase, but gradually they're going to start to unwind and become chromatin again. That cleavage furrow will continue to deepen until eventually the cell is going to split into two separate daughter cells, and these two separate daughter cells are going to go right back to looking like they are in interphase, and they are going to be in interphase because the minute that cleavage furrow deepens and the cells split in cytokinesis, the chromosomes are going to unwind, go right back to that functional form called chromatin. Now, before we end this tutorial, let's do some practice. So what you're looking at here in these two photos is a microscope slide. If you were to use the microscope and look at cells dividing, this is what you would see. You would see many cells all together, and the various stages of the cell cycle and phases of mitosis kind of scattered among the cells. So let's practice picking out the different phases. So first off, let's pick out some interphases. Interphase is so easy. It's just going to look like a cell with a nice round nucleus on the inside. This guy right here is a really good interphase. So this is interphase. Let's see, here's another good interphase, and another good interphase, and another good interphase. Remember that interphase is the longest stage of the cell cycle, so the majority of cells that you see under a microscope are going to be in interphase. You're looking for a nuclear envelope intact and little grainy dots on the inside, which is going to be the chromatin. Now, after interphase, we enter mitosis. Prophase, metaphase, anaphase, and telophase. The key characteristic to all four of these phases is they're all going to have chromosomes, and the chromosomes will be in different areas. For prophase, chromosomes are just beginning to form, so they'll be in the center. Metaphase, lined up in the middle. Anaphase, pulling apart. And telophase, they'll be separated, and you'll see that cleavage furrow at the top and bottom. So let's pick these out. Prophase, we want to see some chromosomes in the middle. Right here, over here in the picture to the right. That is a pretty good prophase. I see the little dots on the inside, and I cannot see a clear nuclear envelope. That's a prophase. Metaphase, we need to see chromosomes lined up in the middle. This guy right over here beside that interphase. This is a pretty good metaphase. I see this set of chromosomes right here forming that line right down the center of the cell. That's a meta. In anaphase, we're looking for the chromosomes to start to pull apart. I bet you've already found it. This guy right here is a really good anaphase. You can see a set of chromosomes here, a set of chromosomes here, and that clear space right down the middle, which tells us they're separating out. And then lastly, we're looking for a telophase. You've probably already spotted it too. This right up here is a good telophase. Here's how I know. I can see a nice cleavage furrow down here at the bottom, a nice cleavage furrow right here at the top, and I see my sets of chromosomes have already separated. So this guy right here is a good telophase. Now, we're not really going to pick out cytokinesis because all cytokinesis is going to be is two daughter cells side by side that are in interphase. So they're going to look just like interphase. You'll just see two cells side by side. Okay, these two right here could be cytokinesis, but again, we're not going to name it because it's redundant. It's going to be two interphase cells. Okay, so we're going to identify, and what you need to be able to do is pick out interphase and then the stages of mitosis: prophase, metaphase, anaphase, and telophase. Look for the location of those chromosomes for mitosis. And cytokinesis just falls at the end. Those chromosomes are going to unwind, become right back, turn right back into chromatin form, and it's going to look like two interphases.